Welcome to the Deep Dive, where we take a stack of sources, articles, research our own notes, and really try to extract the most important nuggets of knowledge for you.
That's the plan.
So today we're tackling, well, a pretty fundamental challenge in biological and clinical research. How do you find and, isolate tiny specific cells from this huge mixed population?
Yeah, it's a big one.
Think about it, whether it's elusive stem cells or those tricky circulating tumor cells, maybe even particular immune cells.
Yeah.
Figuring out what makes them different is just absolute crucial for discovery.
Indeed. And for decades, we've had powerful tools to do this. But recent technological advancements are really ushering in what scientists are calling a new era of cell sorting.
No, new era.
Yeah, this new era combines the incredible detail we get from microscopy with the essential speed needed for modern cell analysis. It's a real step up.
And that's exactly where we're going with this deep dive. Our mission today is to unpack image-activated cell sorting, or IACS. We'll explore what it is, how it's, different from older methods, the ingenious ways scientists have overcome its challenges, and its really transformative potential for everything from basic research right through to personalized medicine.
Hopefully, yeah.
You'll walk away with a clear understanding of this fascinating field, complete with some truly surprising facts about what cells can reveal. Let's explore. So, before we jump into the real cutting-edge stuff, let's maybe look at the foundation. Traditional fluorescence-activated cell sorting, or FACS.
Good place to start. FACS. Developed all the way back in 1969, can you believe? It really revolutionized cell sorting back then.
How did it work, fundamentally?
Well, it works by isolating cells based on the total intensity of fluorescently labeled molecules. So, things inside or on the cell surface that you've tagged with something that glows.
Okay, total brightness.
Exactly, total brightness. It's been indispensable, really.
Yeah.
Think about its use in isolating microbial cells, specific cancer cells, or purifying immune cell subtypes using fluorescent antibodies to compare, like, how much protein is on the surface.
Right, comparing levels.
Yeah, and FACS is also crucial for these big genome-wide pooled perturbation screens. Really helps us understand gene function, disease mechanisms. It's been a workhorse.
Yeah. FACS certainly sounds like it revolutionized cell sorting. So what were its ultimate limitations that eventually kind of pushed researchers to look for new approaches?
Well, while it's powerful for quantifying that total protein abundance, how much stuff is there, traditional FACS completely lacks spatial resolution.
Spatial resolution,
Meaning it cannot differentiate changes in protein localization or distribution within a cell. It's like getting a very detailed count of how many fluorescent flags are on a cell, but absolutely no information about where they are on the cell or how they're arranged.
Oh, okay. So you know how many, but not where.
Precisely. And that significantly limits its applications when you actually need to see more than just a total number. You need the picture, not just the count.
So if traditional FACS couldn't give you that crucial visual information, especially, those subcellular details, what did researchers do historically? How did they cope?
Historically, they turned to, traditional microscopy. That gives you the picture. The challenge was conventional microscopes were great for phenotyping, observing, characterizing cells, but not for sorting them in any kind of high-throughput way. Not fast enough.
Okay, so you could look, but not really sort efficiently.
Right. So the early solution often involved a two-step process. Scientists would kind of couple microscopy with some form of cell isolation, look first, then grab.
What were some examples of these early two-step methods? How did they actually do that?
You'd see techniques like laser microdissection, where you'd image a tissue sample, find your cells of interest, and then literally use a laser to cut them out precisely.
Wow.
Or microraft arrays. Cells are cultured on these tiny little rafts. You image them, find the ones you want, and then isolate them one by one with a microneedle device.
Sounds painstaking.
It was. To get higher throughput, researchers started combining automated high-throughput feedback microscopy with FACS. So you'd image the cells, find the interesting ones, and then optically tag them.
Optically tagged.
Yeah, essentially chemically modify them so they glow differently when you shine a specific light on them. Then you'd run everything through a traditional FACS machine, which could now pick out those tagged cells.
Ah, okay. That sounds like it offered more flexibility, maybe. What were the advantages of doing it in two steps like that?
The main advantage was indeed flexibility. You had great control over the image acquisition, things like magnification, resolution, sensitivity, how many fluorescent channels you used, much more than FACS alone.
And the analysis.
And the analysis was often custom, using advanced machine learning algorithms. These could, discriminate really complex phenotypes much more accurately than someone trying to do it manually.
So if it was so flexible, what was the major drawback? Why did we need this new era you mentioned?
Speed. The significant drawback was speed, just incredibly slow. This two-step process was orders of magnitude slower than FACS.
Really? How slow are we talking?
Well, there's an example in the sources, one experiment involving imaging, analysis, and that optical tagging for 1.5 million cells. Took 8 hours.
8 hours just for one experiment.
8 hours. Yeah. So this made it extremely challenging, really almost impossible, to study dynamic processes in live cells, things that change quickly.
Because the cells would change while you were waiting.
Exactly. Phenotypes could change significantly between the imaging step and the isolation step. So it was largely restricted to fixed, dead cells or live cell phenotypes that were very stable over long periods. Not great for capturing biology in action.
Okay, right. With those limitations in mind, what was the game changer then? What truly combined the imaging and the sorting?
That's where image-activated cell sorting, IACS, finally comes in. The breakthrough is that IACS combines the microscopy and the cells sorting into a single integrated device.
All in one box.
Pretty much. It acquires and analyzes images of cells while they are in flow, as they're moving. This integration is revolutionary because it completely eliminates that critical, time-consuming lag between imaging and sorting.
What exactly is it about putting it all together that makes IACS such a game changer? What fundamental problems did it really solve?
Well, fundamental problems it solved were those huge slowdowns and the inability to capture dynamic changes. By combining the steps, the speed is just drastically increased.
How fast are we talking?
Depending on the specific system's design, sorting speeds can vary. Anywhere from maybe 100 cells per second up to an incredible 15,000 cells per second.
15,000 per second? That's staggering.
It is. And this also means immense time savings.
Yeah.
For instance, an imaging-based genome-wide screen, that example that took 21 days using the old two-step method. It can now be completed in fewer than 2 days with IACS.
Wow, 21 days down to less than 2.
Yeah. It's a major, major advancement for high-throughput applications, and especially for enriching those really rare precious cells that are hard to find.
That is a huge leap. But, okay, getting clear images of cells moving that fast and then analyzing them in milliseconds, that sounds like a monumental technical hurdle. What were the core challenges IACS developers had to actually overcome?
You've hit on them perfectly.
Yeah.
The two main challenges were, first, exactly that. Acquiring blur-free images at such high speeds, these cells are moving incredibly fast.
Right, like trying to photograph a speeding bullet.
Sort of, yeah. And second, performing real-time image analysis. Those images have to be processed, and the sorting decision made within micro to milliseconds to actually tell the sorter, keep this one or discard that one.
Microseconds. Okay, so how did they solve the motion blur problem first? I know some systems, like the Amnis ImageStream, use something called time delay integration, but you mentioned that's mostly for imaging cells in flow, not really for sorting them, right?
That's exactly right. The ImageStream is excellent for imaging analysis, fantastic pictures, but it doesn't sort. The dedicated IACS sorting systems had to develop some really unique solutions.
Like what?
For example, the intelligent IACS or IACS system uses something called virtual freezing fluorescence imaging, VIFEI.
Virtual freezing.
Yeah, it's clever. Imagine a laser beam that scans in the opposite direction to the cell's movement, and there's a synchronized rotating mirror. The net effect is it tracks the cell perfectly as it flies by, essentially extending the exposure time by maybe 1000x for fluorescence without getting blurred.
Whoa, okay, so it kind of cancels out the movement.
Exactly. It effectively freezes the cell relative to the detector just long enough to get a clear image. This allows it to capture both general bright field images like a normal silhouette and two-color fluorescent details.
That's a fascinating approach. And what about the BD CellView system, the ICS one? How do they manage it? They're hitting those really high speeds.
Yeah, ICS uses a different technique called fluorescence imaging, using radiofrequency tagged emission, or FIRE. What's truly fascinating here is that it doesn't actually use a traditional camera like a CCD or CMOS sensor.
No camera. How does it get an image then?
Instead, it uses a line of laser beamlets, each modulated at a different radio frequency. These tag the spatial information as the cell passes through. As the cell moves, the timing of the light emission provides the second dimension, allowing it to reconstruct a 2D image from those tagged signals.
That sounds incredibly complex.
It's very clever engineering. It's a way to capture data without blur at high speed, and it offers 4 fluorescent channels, plus light loss, forward scatter, and side scatter images, so you get a lot of information, similar to traditional flow cytometry, but with added spatial context.
Okay, and the third one, Cosmos, or Deep Cell REMI, how does it handle the imaging part?
Cosmos takes yet another route. It performs imaging a bit more like a conventional microscope, but, and this is the key difference, it captures only bright field images, no fluorescence.
Just the silhouette and internal structure, basically.
Exactly. Focusing purely on the cell's physical appearance, its morphology. By ditching fluorescence, it can actually achieve higher resolution in that bright field image.
So there are clearly trade-offs, then, with these different imaging methods. It's not like one is just better.
Absolutely. There are always trade-offs. While the fluorescent IACS systems like IIACS and ICS gain that incredible speed, they often do sacrifice some image resolution and sensitivity compared to classical, slower microscopy. You can't always see the faintest signals.
Makes sense.
And another important point is that all current systems generate 2D images of cells that are typically rounded up in suspension. This means a cell's orientation as it passes the detector can influence the classification accuracy and the purity of the sort, especially if you're looking for features where orientation matters, like, say, cells in different stages of division.
Right, like if a structure you're looking for is hidden on the backside as it flows past.
Precisely. That's still a challenge.
Okay, so they've figured out ways, clever ways, to get clear enough images at speed. Now for that second major challenge, the real-time image processing and analysis. How do these systems actually decide what to sort in that tiny, tiny window of time? Microseconds, you said.
Yeah, this is where specialized hardware and increasingly advanced algorithms become absolutely essential. A common solution is using FPGAs, field programmable gate arrays.
FPGAs, those are super fast chips, right?
Exactly. They're integrated circuits specifically designed to perform certain tasks like image processing much, much faster than a regular computer CPU because they can be programmed or configured for that single focused job. They do one thing, but they do it incredibly quickly.
So the FPGAs handle the raw speed, but what kind of decisions are they making about the cells based on the images? Is it simple stuff?
It can be. For basic phenotypes, like just looking at cell size or overall shape or maybe total brightness in a channel, simple image parameters are often sufficient. Calculating area or aspect ratio or average intensity, these allow for very rapid analysis and sorting.
But biology is rarely that simple.
Often not. For more subtle or complex phenotypes, maybe the texture of the nucleus or the specific location of a protein spot, that's where machine learning and especially deep learning algorithms become necessary.
Ah, the AI comes in.
Right. Convolutional neural networks, or CNNs, are very commonly used for image analysis here. These algorithms can be trained to discover patterns and categories in the images that are, frankly, unnoticeable or even completely undetectable by the human eye.
So they can see things we can't.
Potentially, yes. They can learn very subtle features. This dramatically expands the opportunities for discovery, letting us sort based on really complex visual criteria.
That sounds incredibly powerful. But
Yeah.
are there any caveats, any downsides to using deep learning for such critical sorting decisions? I mean, sometimes it feels like a black box, doesn't it? You don't always know why it made a decision.
That's a very important question, and yes, there are caveats. The performance of these deep learning methods depends heavily, critically, on the data they were trained on. Garbage in, garbage out, as they say. And yes, as these algorithms get more complex, they can become less human interpretable. It can be hard to understand exactly which features in the image are truly being used for the classification.
So there's risk.
There's definitely a risk that the algorithm might learn to classify based on irrelevant batch artifacts, maybe subtle differences in background fluorescence between experiments, or even tiny variations in how the cells flowed rather than the actual biological features you care about.
Yikes. That could mess up results badly.
It could. So robust training is absolutely critical. You need lots of data, multiple replicates, careful controls, and really good documentation to ensure you're getting high-quality, reliable, biologically meaningful results. You have to validate carefully.
Okay, that makes sense. Now that we understand the sort of underlying tech and the challenges, let's dive deeper into those 3 leading IACS systems you mentioned earlier: IACS, ICS, and Cosmos. Tell us about IACS and Cosmos. Tell us about IACS first, the intelligent one.
Right, Intelligent IACS, or IACS.
Mhm.
This system really marked a new era for high-speed sorting because it was one of the first accessible systems designed to directly use fluorescent image information for sorting decisions in a flexible way. It uses chip-based microfluidics tiny channels etched on a chip.
And how does it sort?
It uses a dual-membrane pull-and-push system for sorting. It's a specific microfluidic actuation method that's significantly faster than using traditional tiny valves to switch flow paths. Its maximum sorting speed is up to about 2,000 cells per second, and as we said, it acquires both bright field and two-color fluorescent images using that VIF tech.
2,000 per second is still pretty fast. And how powerful is its analysis? Can it go beyond just simple size and shape measurements?
Oh yes, its real power lies in its flexibility for analysis. You can use simple parameters like, say, the parameter of a fluorescent signal for basic sorts. But it can also employ sophisticated pre-trained deep learning models, those CNNs again, for identifying very complex subcellular features.
Like what kind of features?
Well, this capability enables sorting of really difficult phenotypes. Things like discriminating between inactive versus aggregated platelets directly from whole blood samples, or even classifying different types of algae based on how a particular protein is dispersed inside the cell. Stuff you couldn't easily do with just total fluorescence.
That's quite specific. Does it need a lot of computing power?
The deep learning part definitely does. It requires significant computational horsepower, often needing computer clusters equipped with specialized processors like GPUs to run the models in real time.
Okay. Is IIACS widely available now, or is it more of a specialized research tool?
It's not yet commercially available as a turnkey system. However, construction protocols for the original version, IIACS 1.0, have actually been published, which makes it accessible to research groups who have the right expertise.
So you could build one yourself if you have the skills.
Essentially, yes.
Yeah.
But it does require a high level of diverse technical expertise, optics, microfluidics, electronics, software, machine learning, to actually build and operate successfully. It's not trivial.
What's its unique potential, would you say?
I'd say its unique potential is in that sophisticated analysis of fluorescent spot signals. Things like chromosome SI spots, which labs use to visualize specific DNA sequences on chromosomes, or maybe P-bodies, which are tiny structures inside cells involved in gene regulation. IIACS can be trained to count these spots, measure their size, or even calculate the distances between spots within individual cells, and then sort based on those criteria. Very powerful for certain biological questions.
Okay, very flexible, but requires expertise. Next up is ICS, or BD CellView. This sounds like the big commercial player that really pushes the speed limit.
It is. The BD CellView system is a commercially available, fully integrated imaging sorter. It's built on a traditional high-end droplet-based cell sorter platform, which is a mature technology.
Droplet-based, meaning it puts each cell in a tiny droplet.
Exactly. And because it's built on that platform, it inherits some key advantages. For instance, it can isolate up to 6 different populations simultaneously in one run. That's a huge advantage for complex experiments where you're looking for multiple distinct cell types.
6 populations at once.
Yeah.
Wow. And the speed.
And it boasts the highest sorting speed of all the current IACS systems, reaching up to 15,000 cells per second. That's comparable to traditional non-imaging FACS, which is remarkable. Plus, it incorporates all the classic flow cytometry parameters, forward scatter, side scatter, alongside its image data from the FIRE technology. Remember, that gives it 4 fluorescent channels plus those scatter measurements.
15,000, that's just hard to fathom. How does its analysis and sort of user control compare to the flexibility of IACS?
Well, the ICS processes images in real time using as pre-programmed FPGAs, as we discussed. These calculate 5 to 6 specific, predefined image analysis parameters. Things like eccentricity, how elongated a cell is, maximum pixel intensity, cell size, radial moment, how intensity is distributed from the center, correlation, and delta center of mass between channels.
Okay, so it's a fixed set of measurements.
It is a fixed set, yes.
Yep.
But what's really powerful here is the real-time user interaction. Researchers can actually see plots of these measured values being generated live as the cells flow through. They can click on dots on the plot, see the corresponding cell image instantly, and then use that visual feedback to draw gates and optimize their multi-parameter sorting strategy on the fly.
So it's very hands-on for the researcher in the moment. They can tweak things based on what they're seeing live.
Exactly. It gives researchers immediate visual feedback and control over their sorting strategy, which is a big plus for many applications.
What has it been used for successfully?
It's been successfully used to differentiate cells from distinct stages of mitosis, for example, efficiently enriching very rare anaphase cells which look different. It's also been used to measure the nuclear translocation of transcription factors, basically, watching proteins move into the nucleus for both targeted and genome-out CRISPR screens.
And did it speed things up?
Dramatically. That genome-wide screening time we mentioned, dropping from weeks to mere hours, that was achieved using the ICS system. It provides truly unprecedented capabilities for certain types of large-scale screens.
So what's the main limitation then compared to IIACS?
The main limitation is that it's currently restricted to that fixed number of pre-programmed image analysis parameters. It does not allow users to implement their own custom machine learning algorithms to look for entirely new, complex phenotypes that aren't captured by the built-in measurements.
Got it. High speed, great interaction, but less flexible on the analysis side. And finally, Cosmos, or Deep Cell RAMI. What makes this one stand out from the other two?
Cosmos, which is now commercially available as the Deep Cell RAMI platform, is functionally quite distinct because, as we touched on, it relies solely on bright field imaging.
No fluorescence at all.
None. This means it works with unlabeled cells and focuses entirely on their morphology, their physical shape, texture, internal structures, visible without staining.
How does it sort? And what about speed?
It uses valve-based sorting on microfluidic chips. Its flow rate is slower than ICS, around 1,000 cells per second, but that slower flow rate is actually an advantage in this case, because it allows the system to capture higher resolution bright field images than the faster systems typically can.
So it's all about what the cell looks like physically rather than what it's been labeled with.
Precisely. And the analysis is where it gets really interesting. It incorporates a large proprietary atlas of annotated cell images and uses deep learning models for real-time, unbiased analysis. It clusters cells based on morphological similarity using techniques like dimensionality reduction.
Unbiased analysis, what does that reveal?
Well, it can reveal subtle characteristics that might not be easily visible or quantifiable by the human eye. Things like tiny blebs on the cell membrane, the presence of vacuoles, pigmentation patterns, internal shading, and texture differences.
You mentioned earlier it could even predict fluorescence.
Yeah, that's one of the most remarkable findings reported with this technology. The morphology contains so much hidden information that the deep learning models can sometimes accurately predict how a cell would have looked if it had been fluorescently labeled, just based on the bright field image alone.
That's incredible. It suggests there's a deep link between form and function, or at least molecular state.
It really does. It's fundamentally reshaping our understanding of how much information is encoded in cell shape. It's uncovering cellular secrets our own eyes, and even simpler analysis methods just couldn't see. This could revolutionize areas like disease detection, where you might not need complex staining procedures in the future. The commercial RAMI system also adds some predefined image analysis parameters area, perimeter, eccentricity, texture to provide more human interpretable metrics alongside the deep learning.
So that sounds incredibly versatile for certain specific applications then.
It is. It's ideal for situations where you have unlabeled samples or samples that are difficult to label effectively, think environmental microbes, or cells from liquid biopsies for cancer detection.
What are some examples?
It's been used successfully to distinguish different cell types based purely on morphology and, importantly, to enrich rare carcinoma cells from patient fluid samples. Another key strength is that its microfluidic chips are sealed, disposable systems. This makes them very suitable for samples requiring sterility or minimal carryover between runs, which is crucial when working with infectious materials or sensitive patient samples.
Makes sense. And the main limitations?
The main limitation, of course, is the complete lack of fluorescence detection. If your question relies on fluorescent reporters, this isn't the system for you. And, as mentioned, its sorting speed is considerably slower than the ICS system.
Okay, 3 very different approaches. Now, I saw a newer system mentioned in the sources, really pushing the boundaries of AI integration even further. Can you tell us about this low-latency AI inferencing IACS? I think it was from Tang et al. In 2023.
Yes, that's a very recent and exciting development. This system really stands out because it essentially replaces conventional image feature extraction entirely with a dedicated AI module that makes the sorting decisions.
So the AI is fully in charge of the decision?
Pretty much. It can perform label-free cell classification and sorting, just like Cosmos in that sense. But here, fluorescent labels were used only for post-sorting evaluation to check how well the AI did. The AI itself made the decision purely based on the bright field characteristics it learned.
That's a huge step forward in relying purely on AI inference. How does its performance measure up, particularly in terms of speed and accuracy?
It's setting some impressive records. They achieved a total sorting latency, that's the time from image capture to the physical sorting action, of less than 3 milliseconds. That includes all the signal processing, the AI inferencing step, and activating the piezoelectric sorter. That's incredibly fast.
3 milliseconds. Wow.
And the deep learning model training time is also remarkably fast, less than 30 minutes to train the model on 20,000 images, which is also reported as a record. And interestingly, it achieves all this with relatively minimal hardware, just one FPGA, one standard PC, and one GPU.
That's efficient, and the purity of the sorting. Given that it's label-free, how well did it distinguish different cell types?
The purity results were quite impressive. It demonstrated 96.6% sorting purity for simple polystyrene beads. But more significantly, for human white blood cells distinguishing different types like granulocytes, lymphocytes, and monocytes, which all play distinct roles in our immune system based solely on their bright field images, it achieved 98.4% purity sorting out granulocytes, 92.0% for lymphocytes, and 89.05% for monocytes.
That's really remarkable purity based just on unlabeled images.
It really highlights the power of well-trained AI to discern subtle morphological differences that allow for accurate sorting without any external labels. It's a glimpse into where the field might be heading.
So with all these amazing but different options, IACS, ICS, Cosmos, and these emerging AI systems, how do researchers actually choose the right IACS system for their specific needs? It really sounds like there's no single one-size-fits-all answer.
That's absolutely correct. There isn't one system that's best for everything. Each IACS platform has unique strengths and weaknesses, and the optimal choice depends entirely on the specific research question you're asking and the nature of your sample.
Can you give us a quick rule of thumb?
Sure. If you have unlabeled samples, or if your primary interest is in morphology-based analysis, maybe looking for subtle shape changes, or if sterility and avoiding sample carryover are absolutely critical, then Cosmos, the DeepCell REMA, is likely your go-to choice.
Okay, morphology and unlabeled samples, Cosmos. What about high speed?
If you need really high throughput, maybe sorting multiple populations simultaneously, and the existing built-in image parameters are sufficient for your needs, plus you value that real-time interactive feedback to optimize gating, then ICS, the BD CellView, truly excels. That's your workhorse for speed and interaction.
Right, speed and real-time feedback equals ICS. And the flexible one.
And then, for situations demanding maximum versatility and flexibility, particularly for advanced machine learning-based analysis of fluorescence signals, or sorting based on those really complex or spot-based phenotypes we discussed, IACS offers incredible potential, assuming you have the expertise to run it or build it.
Flexibility, fluorescence, custom ML equals IACS. Got it. It's clear that the systems are already transforming research. What does this transformative potential really mean for the future of biology and medicine? Where is this heading?
Well, IACS systems will undoubtedly become vital tools for high-throughput screens, drug screens, genetic screens, and crucially, for linking cellular phenotypes, what cells look like or how they behave, directly to their underlying omics data.
Omics, genomics, transcriptomics.
Exactly. Genomics, transcriptomics, proteomics. Being able to sort cells based on a visual characteristic and then immediately analyze their genes, RNA, or proteins is incredibly powerful. ICS, for example, already offers indexed single-cell sorting, which logs the sorting parameters for each individual cell before it goes off for downstream analysis.
Connecting the what it looks like to the what it's made of.
Precisely. These technologies are really poised to become integral, everyday tools in basic life science research labs. And perhaps more excitingly, they hold huge promise for improving diagnostics, predicting patient prognosis, and even developing new therapies in clinical settings.
So looking ahead then, what outstanding questions still remain for IACS? What's next on the horizon as these systems continue to evolve and improve?
There are still quite a few big challenges and opportunities. A major one is resolution and sensitivity. How can future innovations achieve even higher image resolution and better sensitivity to allow visualization of really dim or small structures? Things like low abundance proteins or maybe specific genomic regions labeled using CRISPR-Cas9 technology. Seeing fainter, smaller things is key.
Makes sense. What else?
Also, multicolor imaging. When will publicly or commercially available IACS technologies truly implement robust multicolor imaging using multiple excitation lasers simultaneously? Currently, many systems are often limited, maybe just using a single 488 nanometer laser for fluorescence, which really restricts the range of fluorescent markers you can look at concurrently. More colors means more information per cell.
Right. And what about seeing cells in 3D as they fly by? Is that feasible? Will that become a reality for sorting someday?
That's definitely another frontier. Will real-time 3D imaging become fast enough and computationally manageable enough to actually inform sorting decisions? That could help minimize those orientation effects we talked about, giving us a much more complete view of each cell.
That would be a big step.
It would. And can that machine learning-based analysis, especially the really complex deep learning, be accelerated even further? Can it eventually match the blazing speeds of traditional FACS sorters, like that 15,000 cell second mark, across all types of complex analyses, not just the simpler ones? And maybe the biggest question: how far are we, realistically, from seeing IACS become a routine, ubiquitous tool in biomedical research labs everywhere? And what larger role will it ultimately play in shaping the future of personalized medicine? Lots still to figure out.
We've definitely taken a deep dive today into image-activated cell sorting. We've explored how these really robust, high-throughput systems now allow us to isolate cells based on intricate visual information, fundamentally changing how we approach biological discovery. From the early two-step methods to these integrated powerhouses, it's quite a journey.
It really is. And it's clear that IACS is still evolving rapidly, with developers constantly pushing the boundaries of speed, resolution, color capabilities, and that all-important AI integration. So the final thought might be, as these powerful new eyes into the cellular world become even sharper, faster, and smarter, what new, previously unseen cellular secrets are they going to unlock next?